This is the second part of the lecture notes for Chapter 2, Part B slides, and we are starting with slide number 19. We're going to talk about the four groups of organic compounds, carbohydrates, lipids, nucleic acids, and proteins, but there's a little bit more left, general information. Many organic compounds are polymers. Poly means many, and that means they're long chains of building blocks, and the building blocks would be called monomers. So, and we'll talk about specifically some examples of that, but one that you may already know, the monomers or building blocks of proteins are amino acids. So amino acids are the monomers, and they form chains. And the chain would be the protein, and that would be the polymer. So when polymers are synthesized, the chemical reaction that does that is dehydration synthesis. When polymers are broken down, it's called hydrolysis. There we go. Okay, this is an example of a dehydration synthesis reaction. You've got two monomers. Each one of them has an OH group, and monomer number 1, the entire OH group is removed, and from monomer number 2, just an H is removed, and together, once they're removed, they're combined to form water. So the dehydration part is because water is removed, and synthesis is because the two monomers are now chemically linked by a covalent bond. So an example down here would be glucose, which is blood sugar, and fructose, which is another simple sugar that's found in fruit. When they combine, they form sucrose, which is a disaccharide because it has two sugars. And so dehydration synthesis is the reaction that combines glucose and fructose to form sucrose. And if you look at the reaction in reverse, when sucrose is broken down in our digestive system, water is added. The hydrogen is added to fructose, and the OH is added to glucose, and then that causes them to split apart and become simple sugars, and that's hydrolysis. Okay, now we're going to talk about carbohydrates. The word carbohydrate tells you what elements are in carbohydrates. Carb for carbon, hydra for hydrogen, and then ATE on the end of a chemical name means that it's going to have oxygen in it. The ratio of hydrogen to oxygen is a 2-to-1 ratio. So for example, we looked at glucose, which is chemical formula is C6H12O6. So the hydrogen is 12. There are 12 atoms of hydrogen and 6 atoms of oxygen, and that's a 12-to-6 or 2-to-1 ratio. All right. The monomers of carbohydrates are monosaccharides or simple sugars. We just saw two monomers before. One of them is glucose and another is fructose. They're probably the most common monomers of carbohydrates. Two sugars would be disaccharides, and we just saw a disaccharide, sucrose. And many sugars is a polysaccharide, and a polysaccharide is the polymer of carbohydrates. Polysaccharide is the polymer made up of monosaccharide monomers. All right, so monosaccharides contain 3 to 7 carbon atoms. They're always the monomers of carbohydrates. Some important monosaccharides are the sugars found in RNA and DNA, ribose and deoxyribose. DNA, if you recall, stands for deoxyribonucleic acid. That's because of the sugar deoxyribose that is found in DNA. And both of these sugars are pentose sugars. That means they're shaped like a pentagon or a five-sided structure. And it also means they have 5 carbon atoms. Hexose sugars like glucose are going to have 6 carbon atoms. Glucose is one, which is blood sugar. Fructose, one called galactose, which is found in lactose, or the sugar found in milk. So here's some examples of hexose sugars: glucose, fructose, galactose. Excuse me. They're isomers. That means they have the same chemical formula, C6H12O6, but their structural formulas is a little bit differently. They're arranged a little bit differently. Deoxyribose and ribose are both pentose sugars. That means they have 5 carbons, so they would be C5H10O5. Now, disaccharides have 2 monosaccharides chemically bonded. So important disaccharides are sucrose, which is table sugar, maltose, which is the sugar that's found in malt liquor. It's also found in malt beverages, like if you get a milkshake with malt added to it, malt powder. There's some candy that has malt sugar in it. Lactose is the sugar found in milk, and they're formed by dehydration synthesis of 2 monosaccharides. These are the examples of sucrose is glucose plus fructose combined. Maltose is 2 glucose molecules combined, and lactose is formed by galactose and glucose. Polysaccharides have many monosaccharides linked together in a chain, and most of the time it's a chain of many glucose molecules. Important polysaccharides are starch. We find this is the storage form used by plants, and we find, a lot of our root vegetables like sweet potatoes, regular white potatoes, carrots, radishes. These contain a lot of starch because that's where the glucose is stored in the plant, is in the roots. And then in animal cells, for example, human muscle cells and human liver cells, glucose is stored as glycogen, which is long chains of glucose. And again, it's the storage form of glucose found in animals. This is a picture of what glycogen looks like. It's many chains, and some of the chains branch, but this green molecule is glucose, so it's just long chains of glucose molecules. Now we're going to move on to lipids, which contain the same elements as carbohydrates, but different ratios. So carbon, hydrogen, and oxygen are found in lipids too. But lipids are insoluble in water, whereas some carbohydrates are soluble in water. Glucose is soluble in water, for example. But lipids are always insoluble in water. The main types of lipids are triglycerides, which if you have a blood work done, they'll measure your level of triglycerides. Phospholipids, and I don't know why. I don't know why the H's are capital, except for it was probably like a, what do you call it? You know what I mean? Like the automatic, because they had been using pH in the other slides. But anyway, it should be 2 lowercase h's. Phospholipids are found in our cell membranes or plasma membranes, the membrane that surrounds all of our cells. Steroids are hormones, and eicosanoids are chemicals that are released locally, like, histamine. Triglycerides are called fats when they're solid and oils when they're liquid. They're composed of 3 fatty acids. That's where the tri comes from, bonded to 1 glycerol molecule. And when the fatty acids are joined to the glycerol, that chemical reaction is a dehydration synthesis. Fatty acids are just long chains of hydrocarbons, carbon with hydrogen attached. Main function of triglyceride is energy storage, insulation, and protection. This is what a triglyceride looks like chemically. Here's your glycerol molecule, and here's your long fatty acid chain. And whenever they're joined together, water is removed, just like you saw in combining the two monosaccharides. So it is a dehydration synthesis when a triglyceride is produced. This shows you one triglyceride molecule. And there's an animation I'm going to let you watch, but I do want to explain to you, you've probably heard of saturated fatty acids and unsaturated. If it is a saturated fatty acid, it is typically of animal origin and it's typically solid, okay? Saturated fatty acids, typically animal fats, butter. It's usually a solid at room temperature. And the main thing is that the carbon chain, those covalent bonds that link the carbon atoms are single covalent bonds. And you can see that here. All of these are single lines. You don't see any double lines. So let's go back so you can see one that's got the hydrogens in it. Okay, you see all the way down here. Let's just double check. There's no double bonds between carbon atoms. So this is going to be a saturated fat because I don't see any double bonds between carbon atoms. So that's a saturated fat. And they're just drawing it here without the carbon atoms. If it was a double bond, you'd see a double line. Okay? No double bonds between not even one double bond. Okay? Unsaturated fatty acids have at least one double bond between carbon atoms. It's at least one double bond. That's all it takes to make it unsaturated. And what the double bond does is it causes a kink in the chain, a kink in the fatty acid, so that now that unsaturated fatty acid is going to be an oil or liquid at room temperature. Plant oils like olive oil are unsaturated fatty acids. And then we have trans fats, which are unsaturated fatty acids that have been modified so that they resemble the structure of saturated fats and they're solid at room temperature. They're considered very unhealthy. Normal regular peanut butter that we buy that doesn't separate out into the oil and the peanut part, the peanut paste, uses trans fats. And that's how it gets that texture to where it's a solid at room temperature and no oil sits on top of the peanut butter. But it is very unhealthy. The healthier kind of peanut butter is the natural peanut butter, and it does have a layer of oil on top, but you can stir it in, mix it in, and it tastes pretty good, and it's a lot more healthy, a lot healthier for you. But trans fats make the liquid into a solid at room temperature. And then we have just a note about omega-3 fatty acids. They're very heart healthy. You need to get them in your diet if you can. This is what an unsaturated fat would look like chemically. It would have at least one double bond, and notice how it makes that fatty acid have a kink in it and it bends. It's also going to make that fat into an oil, a liquid at room temperature. So unsaturated are the most healthy, or the healthiest for you. All right, another type of lipid is a phospholipid, and phospholipids contain a glycerol and then two fatty acids. In addition, they have a phosphorus-containing group on that head end of the phospholipid. The head of the phospholipid is hydrophilic, meaning water-loving, and the tail is hydrophobic, or repelled by water. So remember, hydrophilic means it can dissolve in water, so it would be polar. And hydrophobic means that it is insoluble in water, and so it's nonpolar. Let's look and see. The head part is the glycerol, the phosphate, and a nitrogen-containing group. This head part is soluble in water, or hydrophilic, and then the fatty acid tail part is insoluble in water. So what happens is they line up where the heads face outward and the fatty acid tails face inward. So the fatty acid tails never come into contact with the watery environment outside the cell or the cytoplasm inside the cell.